So we've been talking about carbohydrates and lipids. There are two more macromolecules or biological molecules that you need to understand. One is protein, and the other is nucleic acid, which includes DNA and RNA. We are not going to talk about DNA now because we'll learn about it later, but we do need to still talk about proteins. And an example of a protein is an enzyme. An enzyme is an example of a protein. So we'll talk about both of those things in this lesson. Now, proteins are the most diverse of the macromolecules. In other words, lots of different kinds, lots of different functions. These are a few of the functions of proteins. There are lots, and they form enzymes, as I said. They form structures, bone, hair, feathers, horns. They are transport molecules between nerve cells, between cells in the brain and cells elsewhere in the body, hormones. Antibodies are protein molecules. All of the diversity, all these different proteins, are composed of just 20 amino acids. Those are the 20 that make up all proteins. So amino acids are the building blocks of proteins. They are small organic compounds that are composed of two functional groups, the amino group or the amine group, the carboxyl group. On one side of the carbon is going to be a hydrogen atom, and then something called an R group, which varies according to the amino acid. So this diagram up here is kind of a generalized amino acid. Notice there's an amino group on one side here. There's a hydrogen on one side. There's a carboxyl group here, and then you have your R group. So the R group changes. We can look at alanine. And with alanine, our group is methyl. Let's see, aspartic acid, it doesn't really show us, it's probably large. Glutamic acid, again, it's just telling you the R group on this side, but you can see that with each one, the R group's a little bit different. With leucine, we have a whole large piece here with 3 carbons and a bunch of hydrogens. Okay, so these are all different versions of this same basic model of an amino. Now, like I said before, proteins are a chain of amino acids in a certain particular sequence. They are joined together. Each amino acid will be joined together by a condensation reaction, which we talked about before, which is where water comes out. Water is formed in the process. The bond that's formed between amino acids is called a peptide bond. Here is a diagram showing a peptide bond formation. And in this diagram, you should be able to see that the OH group from this carboxyl group here and one of the hydrogens from here join together to form, and then a new bond forms between the carbon and the nitrogen. You still have the oxygen that was part of the carboxyl group, and you also have one of the hydrogens still left attached to the nitrogen. And now you have a bond between them. What's not showing here is, of course, coming off, you have water. So an amine group from one amino acid is going to bond with a carboxyl or carboxylic acid group from another amino acid to form what we call a dipeptide, 2 peptides. Okay? If you have 3 or more amino acids joined together, we are going to now call that a polypeptide. Now, proteins have a 4-level structure. For some proteins, there's only 3 levels, but you need to understand this. Proteins first have what we call a primary structure. The primary structure of a protein is the sequence of amino acids. So we can look at this picture here, and we have a sequence: glycine, proline, threonine, glycine again, threonine again. So I'm reading off here. GLY, PRO, THR. These are the amino acids, and they represent them like they are pearls or beads that are all chained together. So this sequence of these amino acids is the primary structure of the protein. It's the primary structure that is going to determine the rest of the protein. So later in the year when we talk about protein synthesis, remember, it's the amino acid sequence that's going to determine everything else about the protein, because remember, those amino acids are each different in their functional groups, so they behave chemically differently. The secondary structure of a protein is due to hydrogen bonding that might occur between different amino acids. The result can either be what's called an alpha helix, a spiral shape, or a sheet, which is called a beta pleated sheet. You can abbreviate that. The alpha is like this, and the beta is like this. I don't have to know Greek letters, but we have our alpha helices or our beta pleated sheets. So this is our secondary structure. Some proteins will form the alpha helix, some proteins will form the sheet. The tertiary structure, or third level of structure of proteins, is due to interactions between amino acid groups on different parts of the chain. These interactions between mainly functional groups cause the protein to be folded up, and it's this tertiary structure that's absolutely crucial to the function of the protein. Depending on how it's shaped, it's going to behave differently. Especially when we're talking about enzymes in a minute, you're going to see that. So, for instance, in this particular protein, you see it's very large, and in portions of the protein, it's got the beta pleated sheet, and in portions it has the alpha helix. And then due to interactions between these different amino acid groups, you'll get it folding up upon itself. Finally, some proteins, not all, but some do have what's called a quaternary structure, which is your fourth level structure. This is where two or more polypeptides are actually held together into a larger structure. So this is not just one polypeptide chain, not just one chain of amino acids. It's got multiple chains held together by either hydrogen bonding or covalent bonding. And these will make large protein molecules that could be either globular or fibrous. Okay, a globular protein, globular means round, and you could think blobular if you want, globular, it's like a blob. An example of this is hemoglobin. Hemoglobin has 4 different polypeptide chains that are held together with bonds, and that forms the entire protein. Example of a fibrous protein, that would be a long, thin protein, is keratin. You may have heard of keratin. It makes up nails and hair and horns and hooves, and it's also in your skin. Now, something I really want you to understand is the nature of proteins. Proteins, because they have this three-dimensional structure, the tertiary structure, it can be, if it's in a certain chemical situation, an environment in which the chemistry is different, it could be unfolded. We call this denaturation. We say that proteins can be denatured, and this is where the three-dimensional structure of the protein is unfolded. And this can happen because it's often held together by hydrogen bonds, and hydrogen bonds are weak, and they can be broken by things like heat, pH, or salinity, which is saltiness. Something's very, very salty or not so salty, we call that a change in salinity. But this process of protein unfolding is called denaturation. It changes the structure of the protein so its function is altered. Usually that means it can't work anymore for whatever its job is. An example of this is seen with the protein albumin, which is the white of the egg. And you can see the raw egg white there. And before it's heated, it has a particular texture. It's sort of liquidy and slimy. You may have had some experience with raw egg, I don't know, but it's kind of liquidy and slimy. And as it heats, you see the color begins to change and the texture changes until we get all the way down over here where it's been heated for a while, and now it's white and it's firm and it's not slimy anymore, it's not liquid, it's a solid. And so this change that occurs in albumin is an example of protein denaturation, so that can help you understand it. What's happening actually as you heat the egg white, it shows down here how the protein gets unraveled and then formed back again to form this solid structure. Now let's talk a bit about enzymes. Our lab coming up is about enzymes. Enzymes are protein molecules. They are catalysts. And if you remember in chemistry, a catalyst speeds reactions. Now, in the case of enzymes, they speed reactions that would naturally occur in cells and in living organisms, but these reactions happen so slowly, an organism would die before they got the needed nutrient or chemical that they needed. So it's the enzymes that actually make the reaction happen fast enough for living things. What enzymes do as they act is that they decrease the activation energy needed for the reaction to occur. So for every reaction, even if it's a reaction that gives off energy, there is always some energy that needs to be put into it for it to move forward. If the reaction happens rapidly and easily, then it has a very low activation energy. If it's a very slow reaction, like similar to what you would see in living cells, the type you would see in living cells, the activation energy might be really high, and that's why the reaction happens so slowly. So you see in a catalyzed reaction, the second example, the red line, it shows you that in the case of a catalyzed reaction, the activation energy is actually much lower than originally seen. And so that's what enzymes are doing. They're lowering that activation energy, which allows the reaction to proceed quickly in a way that living things can survive with. Notice that there is no difference in free energy between catalyzed and uncatalyzed reaction. So this reaction would happen without the catalyst, without the enzyme, but it just happens more quickly with the enzyme. Now, a couple things about enzymes. Enzymes are proteins, as I said. They are not used up in the reaction, though. Okay? Being a catalyst, they're not used up. So we have the enzyme here. We have the substrate, which the enzyme acts upon. They bind together to form something called the enzyme-substrate complex. And then, when all is said and done, we have two products. In this case, the enzyme helped to break apart that particular substrate molecule. And notice the enzyme is basically unaffected. It's left at the end perfectly functional again. Enzymes are also specific to their substrates, so they have active sites where they fit their substrate just like a lock and key. The newest model of how this works is called the induced fit model. In this example, they don't just fit like a lock and key. Both the enzyme and the substrate are altered during the reaction. So as the substrate, in this case glucose, comes in and interacts with the enzyme, this large blobby thing is the enzyme, it interacts here at the active site or the binding site. After it's interacting, the enzyme actually begins to change shape as well as the substrate being affected. So they both affect each other in the reaction. That's the more recent model. It's not just a lock and key, it's a lock and key that can be molded. Now there are a variety of ways that enzymes work. They might work, it's all chemical. They might work by bringing substrates together that normally wouldn't come together. They might orient substrates so that the reaction can happen. They might weaken other bonds in the molecule through acid-base interactions. Sometimes acid portions, the amine group, or the carboxyl group or the amine group in a base reaction. Sometimes they keep water out, which favors a nonpolar reaction. Now, there are factors that affect the activity of enzymes that are crucial to living things. Temperature, we already talked about. The higher the temperature is, it'll speed the reaction, but if the temperature gets too high, it denatures the enzyme because it's a protein. If the enzyme is denatured, it unfolds and it is no longer functional. This is crucial to living things. pH can also affect activity of an enzyme. Every enzyme has a particular preferred pH and will denature at other pHs. So you might notice here in this graph, we have 3 different enzymes which have highest activity at different pH. We have pepsin, which is the enzyme that's in your stomach. It requires a pH that's very low. Salivary amylase that can be found in your saliva has a neutral pH that it prefers. And then arginase, which I believe is in your intestines, requires a higher pH, and that's why you actually secrete some sodium bicarbonate, I believe, but I could be wrong. We'll talk about that in class. The other factor that affects pH or affects enzyme activity is salinity. Okay, finally, I do want you to understand a little bit about how enzyme action is controlled within living cells. There are cofactors or coenzymes that come in and affect the activity of these enzymes. Sometimes they fit exactly where the substrate fits. If they do that, that's called competitive inhibition, where it comes in and it actually fits where the substrate would fit. And when that other molecule is in there, the enzyme doesn't work. That's a way for a cell to turn off an enzyme, for instance. Sometimes there's something called allosteric control. Allosteric sites are where an enzyme molecule will come in. It doesn't come in actually in the active site where normally the substrate would come in. It comes in another site, and when it does, it changes the shape of the enzyme, and the substrate cannot come in and bind. And that's another way that enzyme activity can be controlled. Also, the cell might control enzyme action through a change in the amount of enzyme it produces. And then also we have these cofactors and coenzymes, which will aid in the enzymatic reaction at times. Sometimes they're vitamins or ions that are required for the enzyme to work. Sometimes we have something called feedback inhibition. This is where the product of the reaction, whatever comes out of the reaction, is actually the molecule that works in the allosteric site, or sometimes the product is a competitive inhibitor. And so that the end of the line, the product that's made, if it builds up too much, it actually feeds back and causes the cell to stop making that enzyme because it really isn't needed. You've got enough of the product. But we can talk about all of this in class. Hope it didn't go too long.